Okay, hi everybody, and thank you for joining us today for today's webinar, Advanced Physiological Signal Processing. My name is Maureen, and I am going to be going through and giving you an introduction on the talk. And also with me is Dr. Dustin Heldman, who's going to be going through some more detailed information on physiological signal processing and how to do so in different types of software applications. Just so that you know, you are able to ask questions during the webinar. On the GoToWebinar toolbar, there is a questions box. Go ahead and type them in there if you have any, and then we will get to those at the end of the presentation if some do come up. Okay, so to get started, what we're going to be talking about today are physiological signals and how to analyze them and how to extract useful information using different types of techniques. So three really good examples of physiological signals or biopotentials are ECG, the electrocardiogram, EMG, electromyography, and EEG, electroencephalography. And these are all electrical signals that are generated from the body. So sorry about that. These are all electrical signals that are generated from the body, and each one of these can be recorded using different types of data acquisition devices. One of those devices is something that we offer called the BioRadio. That is a wireless 8-channel physiological monitor, and using the BioRadio you can wirelessly record all different types of physiological signals, including ECG, EMG, and EEG. All right. So there are several different types of post-processing techniques that you can apply to these types of physiological signals. And there's a lot that can be learned about the signals by plotting them and visualizing them in what's called the time domain. And the time domain is when you are looking at the amplitude of the signal over time. There is also a different domain called the frequency domain, and this is where you're actually looking at the frequency components of the signal that are produced. And both of these domains, or both of these viewing options, can be really useful. So you can learn a lot from the time domain. You can also learn a lot of really interesting and useful information from the frequency domain as well. And what's nice is that with the BioRadio we offer software applications that integrate both the time domain and the frequency domain. So you can view a frequency analysis of your signal, you can look at the amplitude over time. All of that is definitely available. But beyond the BioCapture software, there are several other applications that you can use to actually further this type of analysis and further this type of processing. So moving on to the next slide, this is where I'm going to introduce Dr. Heldman. He is much more versed in these areas than I am. He is the manager of our biomedical research group. So a big part of his job here is to analyze physiological signals. So with that, I give you Dustin.
Thanks, Maureen. So this over here just gives you an example of some of the frequency domain analysis you can do. So this is just one example of a signal plotted that we generated. It's an artificial signal that contains a 5 hertz sine wave and a 20 hertz sine wave added together. And if we do a Fourier transform on this signal and look at it in the frequency domain, you can clearly see the 5 hertz and 20 hertz frequency components. So that's one way of looking at the data. However, something to keep in mind is that the Fourier transform assumes that the signal's stationary. That is, it's not changing, the properties of it are not changing over time. Unfortunately, physiological signals are, of course, never stationary. ECG, heart rate, changes over time. Muscle activity is constantly changing. EEG brain waves, they're all changing over time. So it often doesn't make sense just to look at the entire signal assuming it's all the same. So here's another example, and instead of having a 5 hertz and a 20 hertz sine wave added together, here we plot a plain old 5 hertz sine wave, and then the signal changes to 20 hertz. Now when you look at this in the frequency domain using just a simple Fourier transform, you still see those peaks at 5 and 20 hertz, which shows you those components are there, but it doesn't give you any information on when those frequencies are occurring. So again, the Fourier transform, it's really only useful if you want to know which frequency components exist, but you're not at all interested in when those occur. So there's really a trade-off between time and frequency. So now I'm going to talk about a couple different ways that we can fix this with some advanced analysis, including the short-time Fourier transform and then the wavelet transform, to help overcome some of these difficulties. So I mentioned that the Fourier transform assumes that the signal's stationary, that its properties aren't changing over time. The short-time Fourier transform assumes that the parts of the signal at specific points aren't changing. So, for example, over, say, 1 second or 100 milliseconds, if the signal's frequency components aren't changing significantly in those, we can use this to view the data. And in MATLAB, which is a great tool for analyzing your data, especially that collected with the BioRadio, you can use the spectrogram function to get a visualization of the frequencies and how they change with time. So here's an example of the spectrogram function using that same signal we had before. First, up here on the top left, you see the signal with the 5 and 20 hertz components happening all throughout, and you can clearly see the increase in power in the 20 hertz and 5 hertz range. And then if we look at that other signal where it starts at 5 hertz and then switches to 20 hertz, you can also clearly see this change over time. So we're still looking at time on the x-axis, and then we have frequency up here, and then the color corresponds to the amplitude at that frequency band. So here you see the increases at 5 hertz, and then it changes to 20 hertz over here. Here's a physiological example. This is some EMG data that was recorded while someone was giving a sustained contraction over 200 seconds. The signal looks the same all throughout. However, if we do a spectrogram on it and then look at the median frequency over time, you can clearly see fatigue setting in and how the median frequency starts out around 70 hertz and then gradually decreases over time. Another example is using it to detect seizure activity in an EEG recording. So you can see here there's not a lot going on with this brainwave signal, and then all of a sudden it takes off. And again, if we look at the spectrogram, you can see not a lot going on. And right when the seizure activity starts to occur, you can see these color changes indicating those other frequency components. However, there's always a concern when doing a short-time Fourier transform, in that there's a trade-off between your frequency resolution and your time resolution. What do I mean by that? The better your time resolution, the worse your frequency resolution. So in this example, showing the 5 and 20 hertz signals, you see the increases, the amplitude peaks at 5 hertz and then at 20 hertz, but there's this huge spread. And you can see it goes from about, I don't know, maybe 2 hertz all the way up to 8 hertz. Now, if we shorten, and this is using maybe about 30-second windows, time windows, where you see it every 30 seconds. So if we lengthen those windows to 2 seconds each, you have this really nice frequency resolution, but at the expense of your time resolution. So one way to get around that is using something called the wavelet transform. And unlike the short-time Fourier transform, which has equal time windows at each point, equal resolution in time and frequency, the wavelet transform lets you have separate resolutions for different frequency bands. So you can get that extra resolution at the lower frequencies with a better time resolution, as opposed to having the same resolution across all those. And just to show you how that's done in MATLAB, if you have your signal, you would low-pass filter it and high-pass filter it to split out 2 frequency components. And then you can further decompose it, generally the low-frequency range, into, you cut it in half again with a low-pass and a high-pass filter, and again, And as you keep cutting the signal in half, the frequency components, you can then apply your frequency analysis to those much narrower bands, thereby getting a better resolution where you need it without sacrificing resolution up here where you don't. And just to give a physiological example, this is from a paper where they used wavelet techniques to help remove noise from EEG signals, which was able to help with seizure detection. So finally, I'm going to talk about one other analysis technique called artificial neural networks, which are, it's a type of learning that uses the brain as kind of an example and has artificial neurons that are used to do the computation. So each neuron receives one or more inputs, and then based on the threshold of those, determine an output. And those are really useful for doing mapping and finding different patterns in data. So here's an example. If we have an EMG signal that produces a known muscle force, and we have a whole bunch of these, we can train this network to learn what types of EMG signals produce different muscle forces. And then later on, if we have a new EMG signal, we can use that and predict what type of muscle force would come from that. And this is an actual example from a published paper showing how an artificial neural network was used to do this. So again, these are just a few of the advanced signal processing techniques that I think can certainly help in your analysis, and we'd be happy to take any questions.
Thank you, Dustin. What we wanted to do today was just provide a nice overview of some of the different processes that you can use for physiological signal analysis. So like Dustin discussed, you have your Fourier transform, the frequency analysis component. There's wavelet options, the neural network options. So again, this is more of a top-level overview. If you have more specific questions about any of these, feel free to type those into the questions box. If it's something that we can't get to today, I'd be happy to follow up with you after the webinar to provide some of that additional information. So just going through and addressing some of the questions here. The first is, you mentioned using MATLAB for all of this analysis. What other options are there, and are these options available in your BioCapture software? I can actually field that one, if you don't mind, Dustin. So with the BioRadio, we include the BioCapture software. It's a research application that allows you to collect raw data from the BioRadio. And BioCapture is a very simple and straightforward software package. With it, you can collect your raw data. You can look at data in the time domain, looking at the amplitude over time. You can also look at the frequency domain and look at the different frequency components that are occurring during the signal. And there are a few other functions where you can add annotations, you can use event markers, and you can apply different types of filters such as low pass, high pass, etc. However, beyond that, the analysis functions, they don't really go beyond that. So what we do is we give you the ability to export data files from BioCapture to use them in applications such as MATLAB. We can also support LabView analysis. And another option is a software package we offer called VivoSense. And VivoSense is a more automated analysis package that you can import data from BioCapture into. And while it doesn't go into as much detail as some of the things Dustin discussed, it can provide a really nice overall analysis of some of your data, whether it's looking at changes in your heart rate over time and doing a quick RR interval analysis, or looking at heart rate variability as an example. Looking at the changes in the frequency component of an EMG to be able to detect fatigue. That's another example. So there's a wide variety of options as to what you can use for analyzing your data, and we offer some more canned applications, some canned options, or also that integration with MATLAB and LabView, as well as other Windows 32-bit .NET applications. Sorry if that was a long answer to a short question, now that I realize that. Okay, a next question here. Is all of this analysis post-processing, or can it be done in real time as well? Dustin, you want to grab that one?
Sure. Some of it can be in real time. It depends on what your application is. I know there's some times if you want to use different frequency components as control signals for some sort of prosthetic, that may be useful to do in real time. You can do that using the BioRadio with LabView or with your own programs. A lot of the techniques that you may want to use involve more advanced signal processing than I talked about today. But certainly you can do some of that in real time. And again, a lot of it can also be done after the fact. So there used to be some limitations on, mainly from how fast computers were, about how practical it was to do some of this in real time. But nowadays you can do quite a bit in real time.
Okay. Is there a way to apply different types of physiological signals to these specific analysis techniques? So I can answer a part of that, actually. The signals that we talked about today and the ones about which Dustin gave some detail were the ECG, EMG, and EEG signals. So one component, or one point that I'll mention, is that with the BioRadio you can record several other types of signals, and that includes EOG, electrooculography, or the movement and position of the eye. You can also use different types of transducers for measuring respiration, force, skin temperature, core body temperature, etc. So we were not discussing only those three signals as a limiting factor, but just more in focusing on specific signals for specific applications. And then, several of these different analysis functions can be applied to signals such as respiration or EOG. So a frequency analysis of respiration, if you were doing it, trending it over time, would be a way that you can calculate respiratory rate as one example. Are there any other examples that you can think of, Dustin, for applying these types of analysis procedures to other signals?
Sure, quite a bit. I guess one example might be if you're using a motion sensor. If you're looking at body motion or external motion sensors, you'd want to look at different frequency components. It can be very useful looking at the frequency of, say, different types of tremor that may occur with different movement disorders. And you may also want to see how those frequency components relate to other physiological frequency components, such as the EMG recordings. You can get a lot of information from doing these sorts of analyses.
Okay, perfect. That's actually one area that we specialize in, is data collection and analysis for research and also product development in movement disorders. So Dustin's right, we use motion sensors like accelerometers and gyroscopes for recording the severity of movement disorder motor symptoms. So that's one way that Dustin in his research relates a lot of this back to what we're doing here as a company. That being said, I don't want to take up too much more of your time. Those are all the questions that we have time for. I'll follow up with all of the other ones that have been asked directly. If you have any other questions that you think of, feel free to contact me directly or just follow up with us by phone or by email. So thank you again, everyone, for your time, and hope you have a great rest of the day.